high-potency cannabis used to tie to epigenetic changes, studies suggests. And so a study published in Molecular Psychiatry is the first to suggest the use of high-potency cannabis leaving a distinct mark on DNA, providing invaluable insights into the biological impact of cannabis use. A high-potency cannabis is defined as having delta-9 tetrahydrocannabinol, THC, content of 10% or more. THC is the principal psychoactive constituent in cannabis. The research showed that the effect of cannabis use on DNA in people is different in experiencing a first episode of psychosis, or FEP, compared to users who have never experienced psychosis ever before, suggesting there could be a potential for DNA blood tests to help characterize those who use cannabis, those cannabis users who are at risk developing psychosis, to inform preventative approaches. The senior author for the study, Marta DeForti, professor of drugs, gene, and psychosis at King's College, said, with increasing prevalence of cannabis use and the more availability of high-potency cannabis, there's a pressing need to better understand its biological impact, particularly on mental health. Our study is the first to show high-potency cannabis leaves a unique signature on DNA related to mechanisms around the immune system and energy production. Future research needs to be explored to see if the DNA signature for cannabis use, in particular the one for high potency type, can help identify those users who are at risk to develop psychosis in both recreational and medical use settings. Researchers explored the effects of cannabis use on DNA methylation, a chemical process detected in blood samples that alters how genes are functioning, whether they're switched on or off. DNA methylation is a type of epigenetic change, which means that alters the gene expression without affecting the DNA sequence itself, and is considered a vital factor in the interplay between risk factors and mental health. The laboratory team at the University of Exeter wanted to say that there's 56 authors on this single paper in 30 different institutions. I guess the lead institution was at the University of Exeter in England. Man, that's a lot of institutions. So they're getting basically sampling from all across the globe. So the research also showed that the effect of cannabis use. Oh, I'm sorry, I just read that. The laboratory team at the University of Exeter conducted a complex analysis of the DNA methylation across a whole human genome using blood samples from both people who have experienced first episode psychosis and those who have never had a psychotic experience. Researchers investigated the impact of cannabis use, including frequency and potency, on the DNA of 682 participants. So that's a pretty big field of clinical candidates. So this is in humans they're looking at. The analysis showed that frequent users of high-potency cannabis had changes in genes related to mitochondrial and immune function, particularly CAVIN number 1, the CAVIN gene number 1, which could affect energy and immune response. That's very interesting, just based on what Dr. T was just talking about. So these changes were not explained by the well-established impact that tobacco has on DNA methylation, which usually is mixed into joints by most cannabis users. Well, that statement is just absolutely false. Right? I don't know a whole other than some of my Canadian friends who like mixing their tobacco with cannabis. Dr. Emma Dempster, senior lecturer at the University of Exeter, and the study's first author, said, "This is the first study to show that frequent and use of high-potency cannabis leaves a distinct molecular mark on DNA, particularly affecting genes related to energy and immune function." So the energy comes from it affecting genes that are responsible for the function of the mitochondria. For those of you who don't know, the mitochondria is kind of like the cell's energy factory. So it generates energy by generating a molecule called ATP. And in healthy cells, the mitochondria has to be functioning and has to be energetically favorable conditions for that to happen. So Dr. Dempster meta-analyzed the data from 2 cohorts, the GAP study, which consists of patients with first episode psychosis in South London, and Maudsley NIH Foundation Trust, which I guess was another hospital or something, which consists of patients with first episode psychosis and healthy controls from across England, France, Netherlands, Italy, Spain, and Brazil. I guess that's why they needed all these authors. Again, 56 authors, probably authors from all over the world from where these participants participated from. So this totaled a number of 239 participants with first episode psychosis, and 443 healthy controls represented the general population from both studies who had DNA samples made available. Most of the cannabis use or users in the study using high-potency cannabis used it more than once a week, which was actually defined as frequent use, and had first cannabis use at the age of 16 years old on the average. So, okay, let me kind of break this down. So DNA methylation is actually a natural process in the body where a methyl group, that's why it's called methylation, is added to one of the building blocks of DNA called cytosine. And cytosine is an important building block. It's one of the base pairs in DNA. It's one of the rungs of the ladder of DNA, if you think of DNA as a twisted ladder, which looks like a double helix. So it's an important process that needs to happen to regulate gene expression and gene function, which ultimately regulates protein synthesis and protein function, which is how our bodies metabolize and how our skin is a protein, hair is a protein. So the central paradigm of biology is to take genetic information through the DNA and then through the replication of something called RNA to basically synthesize proteins that we're made out of. And this is basically how biology occurs. So what was known with tobacco users is there are sort of abnormal methylations in DNA, and they tried to relate that to tobacco-related disease. I think they're still in the infancy of understanding that translation of the modification of nucleic acids like DNA and how that relates to through the genetic expression and ultimately protein synthesis, how that affects the well-being and healthiness, and how that affects metabolism during diseases like cancer and immune disorders like AIDS and whatnot. So I'm not saying that this is bad. Actually, they've known for a couple of years, there's a couple previous studies that's been shown that cannabis does impact this DNA methylation process that naturally occurs in our body. What this study shows is that the genes that it does that to is related to immune function, right, which is our body's ability to combat invading species like viruses and bacteria, and also our energy function. And you think of, again, the lazy stoner, right? The lazy stoners, mitochondria might be dialed down because they're low energy. Well, it's not necessarily that simple. But there could be, again, a way to look at these modifications in DNA and see if there's some kind of therapeutic intervention in different disease states. So I think this is very important research. It's very great to be published. It's very great to be able to make the connection between the genes that this happens on and the functions in the body. And so now these researchers will take this, and other researchers will take these results and try to really find out what's happening in the body with high-potency cannabis users. So that's sort of, Dr. Mark's breakdown of this article. I'd be interested in what my cohorts have to say here, but that's it from Friday from here at the grounds of St. Michael's College in Colchester, Vermont. I'd be interested in what my correspondents have to say about this article.
Mark, do you know what other natural sort of processes or things we encounter in our lives that might lead to DNA methylation? And is DNA methylation typically bad? I mean, I kind of would assume any sort of modification to, amino acids and whatnot wouldn't necessarily be good. You're muted, Mark.
Sorry about that, Ian. Yeah, great question. So it's putting a methyl group on the cytosine, right? It's basically, it doesn't change the DNA sequence, but what it does is it creates a structural abnormality that will maybe turn that gene off. So about that for a second. So genes, we know we're trying to tie sort of structural genomics to, say, functional genomics, right? That's where we actually understand functions of these genes. And Ian, genes, I mean, we think we know what they're doing all the time, but they moonlight, they do other things other than what we know that they're responsible for. So it's in that they've tied this now to genes that are responsible for mitochondrial function in the cell, immune response. That's just the very basic, discovery of linking these two. Now the question is could there be therapeutic intervention at the gene level? So we've heard of things that are basic, just gene methods, or looking how do we reduce gene from itself, ultimately expressing a protein that might be, again, in a critical stage of a disease where you don't want to express that protein. Yeah, this is a natural process that happens. It's not like you're making abnormal structures. Body does this all the time. All living things do this all the time, from my understanding. It's just a way kind of turning on and turning off the genes that we need to understand what the physical, biological downstream effects of that are. And I just think we're still in the infancy of understanding, like when you look at an expression of different genes, okay, we could do an induced response panel and we could see that this gene is turned on under this stimulus. This gene is kind of turned off. Well, what does that mean? It's kind of like there's this pull that we need to figure out in between the expression of these genes and how that rates translate to the protein synthesized and modification happen proteins when those modifications happen. I don't know if I answered your but
well, Mark, something's happening with your audio.
It sounded right.
Something's going on with your audio right now, Dr. Mark. Did you
hear me?
Yeah. No, we got the most part. Did everyone get the majority of what Dr. Mark was saying?
Yeah, it just sounded like he was saying it going down a bumpy road.
Okay, fair enough. Fair enough.
Oh, I'm sorry.
I mean, it's
probably our weak internet here.
Yeah, wow. Is it possible that with more research that this could be like they could say that lead to say, hey, this is what creates CHS?
No, this is not a negative.
Well, no.
It's just an indication that cannabis, just like tobacco, influences cells on the DNA level, and that DNA is not static thing. It's dynamic. It's influenced by your environment. There are epigenetic factors. So, it's not just you get a blueprint and that's who you are. Life happens to your DNA too.
Mm-hmm. Fair.
Yeah, so I think where this goes, and I think, again, hopefully my internet's working. Can you guys hear me?
We keep on going, yep.
Okay, so where this goes is an area that's being rapidly expanded now, which is basically gene therapy. And that is, rather than developing an inhibitor for an enzyme, we're going to develop something that works at the genetic level, has to do with the expression of the genes that make that code for that enzyme, right? And so it's a different way of attacking how we could basically intervene in humans. And knowing that this works at the genetic level is going to give researchers clues that, people who have used cannabis for years, I mean, that's what they're talking about as a user group. How is that affecting their DNA? And is that a bad thing? Is that a good thing? Maybe it's a good thing because that methylation that happens then creates a new genetic response that makes us less susceptible to disease. I see Dr. T nodding his head. I like that.
The only thing, Mark, did they ever mention PPAR in that article? Peroxisome?
Didn't mention PPAR.
They did?
No, they didn't mention PPAR, and they didn't really mention the cannabinoid receptors at all. What strictly they're looking at is how cannabis use is affecting these DNA sequences.
Yeah,
and I guess
PPAR is linked to hypermethylation of DNA, so I think they're missing a step.
Okay. All right. So Dr. T
How many doctors
is mentioning what's known as a target. It's known as a target for cannabis and cannabinoids is this PPAR. So maybe that's the missing link. That's the connection between this DNA methylation and what cannabinoids are doing. So yeah, very fascinating stuff.
Sounds like it should have been 57 and added Dr. T in there.
Mm-hmm.